Okay, time to start, I think. So hello, everyone. Thanks for being here today. My name is Maeve Gabrielli. I'm a postdoc at the University of Toulouse in France, and today I'm going to present you some results from my previous postdoc at the University of Ferrara in Italy, where we investigated the genomic consequences of human-animal interaction in the isolated population of Apennine brown bears, or Marsican bears. Okay, so human activity tends to reduce a species range, forcing them to live in small populations. Small populations are characterized by a higher level of inbreeding and genetic drift, which reduces their genetic variability and fitness and lead to even smaller populations in a vortex towards extinction. We can therefore wonder how to avoid extinction and coexist with humans. Some behavioral changes allowing the coexistence with humans have been reported in some cases, but there are few reports of the genomic basis of these alterations. So in this talk, I wonder, what are the genomic consequences of human-animal interaction? Our focal population is the Marsican bear, or the Apennine brown bear, that is a subpopulation of the European brown bear restricted to the center of Italy. So you can see in this map in light gray the geographic range of the brown bear in Europe, and in red is the range of the Apennine brown bear in central Italy. So there are only 50 individuals that live in a very small area of about 50 kilometers that is surrounded by villages. It has a unique morphology, being smaller than the other European brown bears, and it also has a unique behavior. Indeed, it is often found wandering inside villages or in very close proximity to human habitat, houses, but yet there are no instances of aggression and attack of this bear on human, which suggests that it could have a different behavior from the rest of the European brown bears, being less aggressive. A previous genomic study by members of our group analyzed the whole genomes of 5 individuals, including 1 individual at high coverage, and this analysis reveals that it is an isolated population since few thousands of years, and it has a low genetic diversity in general. So you can see on this plot the theta, a measure of genetic diversity across a chromosome, and in red is the genetic diversity of the Marsican bear, and so it is in general quite low, except in some regions that are here in white, where it is as high as the genetic diversity from the other European brown bears. So this suggests that genetic diversity is maintained in some important regions of the genome, such as the regions of the major histocompatibility complex, and so it suggests a role of natural selection in shaping the genetic diversity of this bear. So to investigate the genomic consequences of human-animal interaction, we first produced a de novo reference genome at high quality for the Marsican bear, and we resequenced at high coverage 10 Marsican bears, and we also resequenced 10 brown bears from Slovakia, so in brown in this map, that constitute our large control population. We also used brown bears that were sequenced in another study from Canada and Alaska that constitute 2 additional control populations. And so this talk is structured in 3 parts. First, we will see the population structure and genetic diversity. Second, the demographic history of the clade. And finally, the core of this presentation, that is the inference of selection. So we first aligned our genomes to the reference genome, and then after variant calling, we produced this phylogenetic tree that is neighbor joining a tree based on the pairwise genetic distances between individuals. And what you can see in this tree is that the brown bear from central Italy, the Marsican bear, forms a distinct subclade within the brown bears. And it has a low genetic diversity as revealed by the shorter lengths of the branches compared to the other individuals. So to further look at the genetic diversity, we first looked at the ranks of homozygosity, R or H. So the ranks of homozygosity are regions of the genome that are entirely homozygous due to inbreeding. And so here you can see on this plot the percentage of the genome in ranks of homozygosity for each individual. And so you can see that the Marsican bear has about 60% of its genome that is in ranks of homozygosity. So it's quite high, and it suggests high levels of inbreeding. If you compare it to the brown bear from Slovakia, it has about 20% of the genome in range of homozygosity. We also looked at the size of these regions, and so we observed that the majority of these are quite big, quite long, so they are higher than 5 megabases in the Marsican bear, which suggests recent inbreeding, as these regions have not been broken up by recombination yet. We then looked at the genetic diversity at the genomic level, so looking at theta for each individual. You have the theta of the Marsican bear here in red, and you can see that it is much lower than the diversity of the other individuals, or the other brown bears, except maybe for the Spanish one here. Okay, this is dying. That is the yellow point that you can see that is known to be quite an inbred population. But quite interestingly, if you look at this theta outside the range of homozygosity, you can see that the genetic diversity is quite similar between the Marsican bear and the rest of the European brown bears. So suggesting, like in the previous study, that genetic diversity is maintained in some regions of the genome. We then investigated the demographic history of this clade. So first looking at the ancient history from the PSMC method. So the PSMC method allows you to reconstruct the effective population size over time from one diploid individual, one diploid genome. And so in these plots, I show you the result for the two individuals with the higher coverage for the Marsican bear in red and for the European brown bear in brown. And so in this analysis, so be careful, the past is on the right, you can see that there has been a population decline in the Marsican bear about 3,000 years ago. And so this decline corresponds to the time of arrival of human in the region. So this suggests that when humans arrived in this region in Italy, they constrained this bear to this very small region in the center of Italy. And so that's why we observe this decline in the population size that we do not observe in the European brown bear from Slovakia. Then we also wanted to reconstruct the more recent history, and so to do that, we used another method called GON. So GON allows you to leverage the linkage disequilibrium information at the population level in order to reconstruct shorter time scale, so we can reconstruct the effective population size of the last tens or hundreds of generations. And so in this analysis revealed a slow but constant decline over the last 1,000 years for the Marsican bear here in red. And in contrast, we were quite surprised at the beginning to observe this decline in the brown bear from Slovakia, that is about 400 years ago. But then we understood that it corresponds to the end of the medieval ages, and we know that it is a time when the brown bear was heavily persecuted in Europe, so that could explain this signature that we have here with this analysis. But what is important is still now in the present, the brown bear from Slovakia has a higher effective population size than the brown bear from central Italy, the Marsican bear. So we have this Marsican bear that has a low genetic diversity and that has a low population size since quite a bit of time. So we were wondering, how can we infer selection despite high levels of genetic drift, as selection and drift can lead to confounding signatures. So to try to tell them apart, what we did is that we used multiple and independent approaches, and we also used multiple comparisons to try to detect the signatures of selection that were unique to the Marsican bear. So we first used race at the level of single population, then XPCLR between pairs of populations. So XPCLR allows you to compare the allelic frequency differentiation between pairs of populations and accounting for genetic drift. And finally, PBS for multiple populations, so population branch statistics. In our case, four populations, so the Marsican bear, brown bear from Slovakia, Alaska, and Canada. And so for the focal population, we used the Marsican bear, obviously, but we also redid all the analysis using the Slovak bear as a focal population to see if we had the same genes that were popping up, and the answer is no. So we were quite happy and confident that the signature that we detected was unique to the Marsican bear. And so these are the results that I'm going to show you now. So for these outlier genes, we performed then a gene ontology enrichment analysis, and we also just looked at the function of these outlier genes. So you can see in these three Manhattan plots the three analyses that we run with the Marsican bear as a focal population. And so in green are the top 1% outlier genomic regions. And so we then extracted from these regions a total of 500 genes under putative selection in the Marsican bear. We then performed a gene ontology enrichment analysis, and we saw that these genes are involved in pathways related to developmental process, anatomical structural development, and multicellular organism development. So these pathways are related to the change of morphology with the Marsican bear. And quite interestingly, we also found some pathways that in humans are related to over-friendliness and social disinhibition. So this suggests a change in behavior in this Marsican bear that could have been selective. And in link with that, we also identified 20 outlier genes that are related to tameness evolution in domesticated animals. So these are the genes that are involved in these plots. So for example, GRM7 is a gene that is associated to less stress and less aggressiveness in the domestication process of the sheep and the pig. So all of these results suggest a behavioral shift towards tameness in the Marsican bear, and we hypothesized that this could be due to an indirect selective pressure exercised by humans that have been living in close proximity to the bear for thousands of years. So this could be a case of incidental domestication of the bear. So to wrap up, to look at the genomic consequences of human-animal interaction in the Marsican bear, we compared the genomes of Marsican bears to genomes of 3 other control populations. And so we first observed a low genetic diversity and high inbreeding levels in the Marsican bear that was characterized by a demographic decline 3,000 years ago, followed by a more recent reduction in the last 3 centuries. And finally, we could evidence some signatures of positive selection on genes associated with behavior that could explain the tameness of the bear. So these results have great implications for the conservation of this iconic group in the central of Italy. So with this, I would like to acknowledge the endemic seed group. So it was part of a bigger study on the conservation genomics of 5 Italian endemics, involving a lot of different universities from Italy, and involved all the members of the groups that were involved in this study. And with this, I would like to thank you a lot for your attention. So we have time for a few questions if you have some. Yes, please. So yeah, we think it was about like the same timing or so of the bottleneck, so around 2,000, 3,000 years ago. Yes?
Do you see strong morphological differences in the Japanese bears?
There are some, yes. It is like in general smaller than the rest of the other bears, and also the cranial morphology is different, and it has a bit of a different diet, being a bit more herbivorous than the other bears.
And do any of these differences correspond to the domestication syndrome that you see in domestication?
Yeah, the problem is that we don't have in our dataset this morphological information, only the genomics. Yeah. But that would be interesting to see that.
Okay, I think I'll just get started. So I'm Rishi De-Kayne. I'm a postdoc at the University of California, Santa Cruz. And today I'll be talking a bit about the unpredictability of sulfide adaptation across these pusillid fishes that I've been working on. And thank you all for making it through the conference so far and to the sort of fabled Adaptation 14 that I know you all wanted to be giving a talk in today. So I think, like many of you, I'm motivated by sort of a host of different evolutionary and biological characteristics of organisms and the links between these different features. So things like various aspects of phenotype evolution, be that physiological traits or behavioral traits, things like fitness. So how are these traits passed on? What's the sort of heritability of these things? And how these link to the basis of this variation, things like genotypic variation that you see here, like this SNP. And I think the interactions between these I've seen in all of the talks this week is kind of something that motivates all of us. I have to say I feel a bit silly because this is the first conference I've ever been to where there's been maybe 50 plus structural variant talks, and I've actually spent the last 2 or 3 years mostly focusing on structural variants. Today's talk won't be about structural variants, but I have to just take a short moment to plug a paper that we just, or preprint we just uploaded, which is a product of my previous postdoc on structural variant evolution in these butterflies. And so just for 30 seconds, I'm just going to say these butterflies in Africa that we were studying have this amazing structural variation that underpins wing pattern variation. And there's this amazing role of what we call like incomplete recombination suppression, where we get this allelic turnover that I think is really exciting. And I hope if you have some time after the conference when you've recovered, I really encourage you to go and have a look at this paper. But the paper I'm actually going to talk about today is this one that we published on percillid fishes, which have this amazing adaptation to sulfidic environments. And in this case, I'll mostly be focusing on this phenotype-genotype link. And one thing that I think we're all aware of, especially after all these talks, is that these various aspects of variation across levels kind of contribute to this phenotype that we're interested in. So single nucleotide polymorphism, structural variation, epigenetic variation can all have these kind of combinatorial effects, if you like, through gene expression, through to phenotypic variation, and also directly. So these things can all contribute to the end trait that we find fascinating. And in this case, the fascinating trait is the sulfide adaptation. And these percillid fishes live in these environments across Mexico and in other places where you see the stream on the left is a non-sulfidic stream, a normal stream, whereas the stream on the right has this high concentration of hydrogen sulfide leading to a number of environmental differences. You can maybe see from this image that the water is very cloudy. So that is not the only change. The abiotic environment is very different. And also the biotic environment is remarkable in the sense that there's a high concentration of bacteria in these water and sort of other extremophiles in high abundance. And this poses a bit of a challenge for fish because, as you might imagine, fish don't really like being in really toxic water. And this specific adaptation is a little bit confusing in the sense that most organisms produce hydrogen sulfide endogenously because we use it for cell signaling. And so actually to adapt to these kinds of environments requires this sort of paradoxical process of kind of modulating your own endogenous hydrogen sulfide production while accounting for this quite toxic sort of environment that you find yourself in. And adaptation to sulfide is not just found in 1 or 2 lineages, but it's actually found across the percillid clade. So all of these clades with an arrow have evolved sulfide adaptation of some kind. In some cases, this is limited to a population of that species, which lives in a sulfidic environment and kind of thrives there. In some cases, in red, actually all members of that lineage are sulfidic and have evolved and diverged as a result of that adaptation. And from this, it's been this kind of putative thought that sulfide adaptation has evolved independently in these lineages. As you might imagine, as biologists, we often think, well, is this adaptation convergent, or to what extent is this convergent? And what can we learn about convergence and the repeatability of evolution from these kinds of systems? So some studies have already looked at this from a variety of different angles. So one of them, for example, here is about gene expression. This tree that I'm showing is a species tree between these different taxa where sulfidic lineages are indicated with these yellow branches. And people looked at gene expression patterns across these different taxa. And what they find is that actually when you look at gene expression and compare individuals clustered by their gene expression profile, that sulfide adapted lineages actually cluster together when we look at that gene expression pattern. So there's some kind of convergent patterns of gene expression, which you might expect. They have a shared environment and they have many shared adaptive traits. And there's also some evidence when we look at coding sequences and amino acid substitutions that sulfinic lineages share some convergent amino acid shifts. But the degree to which this kind of pattern of convergence is shared across other biological scales, and especially if we step away from coding regions, whether there's variation we can pick up there, has been an ongoing question. And so for this study, I'll be kind of breaking down these aspects of variation into coding regions, promoter regions, and enhancer regions, where we've tried to get a bit of a handle on the pattern of convergence across this clade as a whole. What I mean when I talk about these in the talk, coding regions is things like exons, so we'll be having sort of like gene alignments from exon sequence. We'll have alignments of promoter sequence, so that's 1 kilobase upstream, and enhancer regions, which we identify with this cap small RNA-seq, which is very complicated. I'm not going to go into it, but if you buy Blair a drink at the social later, he will happily explain it to you, I'm sure. And it's a really amazing method for capturing enhancers that are much further away from the gene of interest. So in total, what we did is we took 7 sulfidic and non-sulfidic lineage pairs that are shown here. We sequenced them with short-read sequencing. We produced a species tree from over 21,000 genes. And then the idea of this analysis was to compare the species tree with trees from other regions of the genome. And we use this software, RER Converge. And the nice thing about this software is it gives us this kind of expectation where we're able to identify convergent regions of the genome that are evolving convergently faster or slower in our focal taxa, which in this case are the sulfidic lineages. So if you imagine we find a region of the genome with a tree like this on the right, in this case, you can see that the branches leading to our yellow individuals are shorter than those of the blue, and we would kind of deduce that this is a tree in which this region is evolving slower in sulfidic lineages than non-sulfidic, and these kinds of trees are indicative of purifying selection. And in contrast, where we find that sulfidic lineages have these longer branches leading to them, these might be areas where we see directional selection. And this kind of denotation of like negative rho for ones evolving slower in sulfidic lineages and a positive rho is the metric that RER Converge kind of gives us to work that out across the genome. And then, yeah, it's this comparison between these trees, which is kind of the key part of this analysis. So starting with the species tree, what we find is that indeed we kind of recapitulate this idea that taxa are not segregated by whether they're sulfidic or not. So you see at the top we have Gambusia species where we have non-sulfidic and sulfidic as sister lineages. We have Siphophorus, which you might recognize, these swordtails, which have a whole host of amazing evolutionary biology, all the way down across our taxa. And when we look at variation, because the way this analysis works, there has to be a certain amount of variation within these different windows. So you'll see the numbers are a little lower than in the previous slides. But in total, we analyzed 19,000 genes, around 15,500 promoters, and around 8,500 enhancer regions. And in each case, what I'll be showing is whether we see relative evolutionary rate shifts that are higher in sulfidic lineages, indicated in red, or lower in sulfidic lineages in gray. And so they look like this. So for genes, we see a kind of much higher number of genes evolving slower than we would expect in sulfidic lineages, 258 compared to 17 that are evolving faster. Similarly, in promoters, there's this huge disparity between the two, with 12 evolving faster and 142 evolving slower. And enhancers is the only one where it's kind of close and actually flipped the other way. So we see 20 enhancer regions that seem to be evolving convergently faster than we expected, and 7 slower. And so in total, we find many shifts for the number of genes that we looked at. And I should say, I'm not going to go into it, but the significance of these convergent shifts is determined by a kind of permutation test where we essentially shuffle the labels as to whether a branch is sulfidic or not. And so just to give a visual representation of what this looks like, this is a tree that represents a region of the genome, or a gene in this case, evolving faster than we expected. This is actually the most extreme positive rho value that we see. And you'll see that while there are a number of sulfidic branches that are much longer than their kind of sister non-sulfidic branch, the nice thing about this analysis is that it captures convergence in a way that is not super stringent. And so actually convergence in this sense is not a complete convergence across our tree, which I think is a nice way to look at it. And in contrast, what you see here is a region where there are non-sulfidic branches that are longer, and this region of the genome has a negative rho of 0.6. So one of the things that you might have found striking immediately is that we do find more instances of reduced evolutionary rate in sulfidic lineages than positive. I'm not quite sure why this is, but I think we've tried to interpret a little bit as to why you might have a kind of conserved function of these regions that obviously if you allow regions to just kind of diverge, you're probably going to die if you're a fish in these streams. And what we were then able to do is kind of compare these regions that we found with published lists of either genes that have been shown to be involved in sulfide adaptation or genes that have been shown to have this differential expression pattern. So the first one is a list of sulfide detoxification genes and oxidative phosphorylation genes from Tara Ryan's paper in 2023. And these genes are all involved in mitigating the effect of hydrogen sulfide on mitochondria, which is the main way in which it actually inhibits respiration in fish. And we find a number of genes overlapping. All the genes that overlapped with this kind of set of putative genes evolved slower than expected. But one thing we notice is that enhancers have this kind of mixed signal where some evolve faster, some evolve slower. And I think that makes sense when you think about coding sequence and transcription, that a nice way to modulate the way in which you respond to an environmental stressor is through promoters and enhancers and regulatory reasons rather than coding sequence itself, which has kind of a narrow room for error. And then when we look at genes that are differentially expressed in sulfidic lineages or across different taxa, we again find a number of overlapping genes. And we obviously had to do the kind of classic thing of looking for ones with an interesting kind of story behind them. But the reality is that some of these genes are involved in hydrogen sulfide modulation within organisms, so things we know are essential for maintaining basic cell signaling and metabolic processing. We also have genes that we know are involved in sulfide detoxification, including G6PD, and some of these other genes that are really important for reducing oxidative stress. And then again, we have some other candidate regions where people have done detailed work looking at what happens when you can't actually reduce oxidative stress. And one thing I would stress about the study is that actually one of the challenges is we don't have a great annotation for these taxa yet, and so actually we're using human orthologs here. So if any of the genes kind of catch your eye for being weird in your study taxa, I would be curious to hear what you know about them, because yeah, we've been a little bit hesitant to make too many strong links to some of these annotations, as you might expect. So in conclusion, we find many rate-shifted genes, promoters, and enhancers, and I think the study kind of captures the importance of looking more holistically at these kinds of things, looking at adaptive stories. Most of the changes we find are actually negative, perhaps indicating that they have this conserved function. They're essential for these fish to survive in these streams. One of the things I didn't go into too much detail is that actually, in many cases, we had a gene and its promoter included in our analysis. We were able to look at, do we find both a gene and its promoter shifting significantly? But actually, we found very few examples, only 3 examples where a gene and its promoter had a convergent shift. And I think, again, that kind of makes sense when you think that if you're evolving, you don't really need to tweak both of these things, both your core kind of coding sequence and the promoter. That just tweaking one can be sufficient for having these adaptive shifts. We find a number of genes that overlap with these known genes that are important for sulfide adaptation. And generally, I think better understanding the repeatability of evolution by stepping outside of coding regions is kind of a key take-home. So with that, I'll just say thanks to my co-authors and collaborators and the Kelly Lab at UCSC, and thanks for your attention. I'll take any questions you have. Yes.
Do you have an estimate of how many of these regions were found to be convergent in all the lineages versus just some of the lineages?
Yeah, so the tree I showed was actually the most extreme either end. So I think it's safe to say that basically none are convergently shifted in all taxa. We sampled quite a wide range of taxa, so I wasn't that surprised with that. But I think it definitely changed the way I think about these analyses of convergent evolution, in the sense of how wide you define the net is kind of whether or not you're going to find convergence is kind of tightly linked to that. So I think, yeah, not many instances across 7 independent genera. Yep.
Did you see different functional classes of genes evolving at the protein coding level versus the regulatory level?
Yeah, so one of the things I really expected to find, if I'm being honest, from other enrichment analyses I've done, was some kind of convincing and hopefully something tied to sulfide adaptation. But actually, we found very few kind of ontology classes that were enriched at any level. And the ones that we did find, I didn't present because, frankly, it's a bit of a tenuous link to kind of link them to anything to sulfide adaptation. So no, we didn't. But I think, yeah, I don't know why that is. But I'm guessing it's just that this environment is challenging for many different regions across many different kind of classes of genes and different functional categories. And so, yeah, thanks. There we go. Thanks for your attention. Appreciate it.
Let's get started. Thanks for your patience, and sorry about that. My name is Maeve Stokes. I'm faculty at Florida State University, and we're going to take a slightly different flavor of perspective on adaptation today, and I hope you'll enjoy it. So I want to start by reflecting on how adaptation may play a different role in the diversification of freshwater fishes and landscapes that are organized into different hydrologic units. So the role of adaptation in the diversification of freshwater fishes and lakes has been widely reported. We have our iconic adaptive radiations in African cichlids, ecological speciation of sticklebacks in post-glacial lakes, and I'm going to try to go fast, so bear with me. And here we're looking at a landscape that's physically organized in a fundamentally different way, and this is what I think about a lot. I'm originally trained as a geomorphologist. So here we're looking at a topographic map of the Allegheny Plateau in Pennsylvania, and you can see that this landscape is organized into these different hydrologic units, this beautiful dendritic river network. And the role of adaptation in riverine fishes is less clear than in lake fishes. These fishes are sister species, and riverine fishes in North America are almost exclusively allopatrically distributed, and most speciation studies have focused on historical biogeography, including my own work. So here's a very brief introduction to an example of that kind of work. Here we're looking at the distribution of the greenfin darter in the upper Tennessee River in Tennessee and North Carolina. This species is distributed almost exclusively on metamorphic rock. And in this work, which I will tell you about in 2 slides, we show that erosion through the different kinds of rock on this landscape exposed different rocks at different times. And in doing so, isolated lineages of this group in the headwaters of the Tennessee River. And in doing so, it created divergent but ecologically equivalent lineages, and I mean that both in terms of the environment and the morphology. But we think that we have a rare example where we see some evidence of the role of divergent ecological pressures and selection within this group. And you can see very clearly that they look differently. They have different snout lengths. We have our long-nosed darter and our short-nosed darter. In allopatry, we may have an absence of selective pressures, similar selective pressures, or different selective pressures. And we would expect some divergence along morphological traits, environmental conditions, and there should be some genomic basis to that today, which I don't have results for you today. So for all the genomics people here, wait a few years. We're interested in this as a way to explain the diversity of the southeastern North American temperate freshwater biodiversity hotspot. So here's our study system. We have 3 species, as in the guidebook, Persina foxocephala, Persina nasuta, which is our long nose, and Persina Bruce Thompson and I were looking at a map of rivers in the central United States. The taxonomy is not adequate to explain the diversity of the system, and it gets complicated. So throughout the talk, I may say nasuta from the Arkansas River system. What I mean is the long-nose morphotype from the Arkansas River system. And I'll try to make that as clear as possible with these red and blue dots. But as you'll see here, the phylogeny is a little complicated. We have 2 instances where red and blue dots are sister to each other. And I'll move on to the next slide and walk through these bit by bit so you can take a better look at their geography. So here we have foxocephala and nasuta, both from the Arkansas River system. In the middle there, it looks like they're sympatric, but in reality, there's only one creek where they occur together, and we have evidence of a putative hybrid. This is exciting because this is one of our only examples of sister species and darters living in the same creek. Here's another example of where we have foxocephala and nasuta as sister lineages, allopatrically distributed. And then for our final comparison, we'll compare 2 nasuta type. Okay, so we're going to focus on these 4 divergence events and try to understand whether there is ecological and morphological evidence for divergent niches. So let's start with snout length. The whole reason that we have these 3 taxonomic buckets for these species. In 3 out of 4, so the stars mean we see a significant disparity in snout length. In 3 out of 4 of the comparisons, we do see a difference in snout length. The greatest differences are within the Arkansas River system, and then the bottom between the St. Francis and Black River systems. Okay, I wanted to know whether there was some environmental way to predict these difference in snout morphology, so I collected environmental data from the StreamCat data set from the EPA. So these are variables associated with the streams themselves. We characterize the background environment as well by subsampling these flow lines from within the watersheds where these species are found. And so we have variables that characterize hydrology, climate, land cover, and the substrate of the streams. So here are PCAs. We again see 3 out of 4 of the comparisons have significant differences between where these species are found, but because these are in allopatry, we can't just say where they're found is different because the background environment is varying as well. So our null hypothesis is that this disparity reflects the variation in the background environment. And so in this PCA here, we have the background environment, and the big circles and the dots are where we find the species. If the difference in the background environment is less than the difference between where we find the species, we have evidence of divergence. If the opposite is true, we have evidence of conservatism. And if we don't see a difference between the variation in the background environment and the occurrence data, perhaps there's evidence of neither. We do this for all the PC axes. Here I'm just showing the first 4 principal component axes, and we see evidence for niche divergence in 2 out of 4 of our events that we're comparing. The first and the third, if you remember all of these river names, I'm very impressed with you, but these are our 2 systems where we do see significant differences in the snout length, so that's great. Okay, so 2 pairs exhibit evidence for niche divergence and 2 largely conservatism. What are these divergent environmental conditions? The long-nose type tend to occur in rivers with more sand, more precipitation and higher temperatures, more forest, lower elevations. How is that all connected to the way a fish makes its living and may adapt to require a longer, shorter snout? We think the answer may be in the diet data. So my colleague Rich Harrington from South Carolina DNR has collected gut content data from museum specimens. So in white, we have Foxocephala from the Arkansas, and gray, the longnose from the Arkansas. And the take home is that our longnose don't eat many chironomids, which is rare for darters, and they have a higher proportion of Plecoptera and Odonata. We see a similar pattern in this system, and we can put this into multivariate space and quantify the difference between our comparisons that we're interested in. So the bottom now is the 2 multivariate space axes and the absolute difference in this diet composition. And so again, our Arkansas River system, where we have the largest disparity in snout length and the greatest difference in the background environment, is where we see the largest difference in diet. So we think diet is the link between the environment and the morphology. So putting this all together, we do see a disparity in snout length in 3 out of 4 of our comparisons. We see a significant environmental disparity that exceeds that of the background in 2 out of 4 of those. And we think that diet could help us link the environment to the morphology. So some more information for you. The third comparison here, they have the oldest divergence age. They have high meristic trait variation, which is used to delimit species in freshwater fishes, but it's maybe more neutral traits, and they have high genomic divergence. And so I did this even despite the delay, so thank you for listening to my fast talking. I think that what we have here, we have different outcomes of allopatric speciation, possibly the role of adaptation within a group of darters, which is an exciting new discovery for us in this group of North American freshwater fishes. I think that we may have a gradient in selective pressures across these environments, and that diet is the link between environment and morphology. If you're interested in this kind of work, or more broadly incorporating geologic data into your biogeographical studies, please do get in touch with me. Thank you.
Okay, I'm going to start. Thanks everyone for being here today. I'm Fabian Salgado, last year PhD student from the University of Melbourne, and I'm going to tell you today about spiders and colors. So ecologists and evolutionary biologists usually look for marker phenotypes to understand how variation change in time and in the geography. Some examples are the famous beaks of the Galapagos finches, or for example, these growing ecotypes of this sunflower. Usually, we select these marker phenotypes because they are easy to follow in time and in the geography. One of the most famous marker phenotypes is coloration because we know how to measure, we like colors. And usually when you have multiple color forms in a single population, it's defined as color polymorphism, and the study of color polymorphism has improved our understanding of how natural selection operates and drives the evolution of traits. Color polymorphism has been mainly studied in lineages that use coloration as a sexual trait or as a signal of aposematism and warning for predators, but we don't know much about what is happening in other organisms that don't use coloration for this. So an example of these are spiders that use color to reduce predation via camouflage, possibly via aposematism, because they are ectotherms, they rely on coloration to reach certain environments, and the ones that build webs, because they are zero-point predators, they usually use colors to deceive their prey, resembling things that are around them and capturing them in the web. So in a systematic literature search, I study how many times color polymorphism occurs. So this is a phylogeny of the two main families of well-building spiders. And as you can see, color polymorphism is quite common in these groups. I found 140 reports of color polymorphism, and we actually don't know much about this. An example is like 38% of the descriptions in both black and bright morph, but only until 1915 we knew that black colors in spiders are caused by melanin, something that is really known in other organisms. So that means that the mechanisms behind color polymorphism in spiders are largely unknown. So to answer this question, I decided to study the Christmas spider as a Acanthaminax. It's a great model to study this because it's widespread across multiple environments, it's really abundant, easy to find, and also really, really cute. This spider has a color polymorphism, so you can find females that look black and white like the one at the top, or completely black like the one at the bottom. This is how they look in reality. We don't know much about them. So in one of my chapters of my PhD, I studied like the color proportion as color variation through the geography. So this is in generality what I found. So the black and white morph is really abundant, and you can find it in mostly all the locations. But when you find the black morph, it's in a lower frequency that tends to increase towards the coast. That is where there are higher values of precipitation. So when you explore this for real, you find that the black and white morph proportion in the field doesn't change with precipitation, but the black morph actually increases its frequency towards places with a higher precipitation. If someone wants to talk more about this in a bit, we can talk after the conference. So with ecology in mind, we decided to ask ourselves what are the genetic mechanisms that generates dark colors in spiders. So the main difference that you can notice between these two color morphs is like one possibly has more melanin than the other. But when we look at these phenotypes using cryo-SEM, we notice that the black and white individuals has these structures that look like caves. And when you zoom in, you notice that inside these caves there are these little crystals, guanine crystals, that are responsible for the white coloration of the spider. So with this phenotypic difference in mind, if we explore the gene expression profile of these two morphs, I expect to find that the black and white individuals would be more close to each other than to the black individuals. And also, I expect to find differential gene expression in genes associated either with melanin development or guanine crystal development. So to test these predictions, I first observed how these spiders change color through the development. So we collected multiple egg-sacs in a locality near Melbourne. I took them to the lab and I grew them. So at first, when the spider hatch, they are all brown. and tiny, as you can see on your left. After 7 days, they all have this black and white pattern with the white being at the top. And after 40 days, you can notice which phenotype they are going to have when they're adults. And after 90 days, they fully develop the phenotype. So we decided to get RNA of individuals at these 3 stages. The first one as a reference, the second one because it's the first stage where we can identify the phenotypes, and the adults because it's when the phenotype is fully developed. So we assemble a reference transcriptome for the species, we annotate the transcriptome, and we explore for differential expression. So I'm gonna show you some of these preliminary results. So when we look at the PCA, using the top transcript differential express transcript, we recover the 3 stages of development that I use. So you can see that the little babies are on the right, highly differentiated. Then we have the juveniles at the top. We couldn't see any difference between black and white individuals. And at the bottom, we have the adults that show a clear pattern of differential gene expression between the black and white. So I'm gonna talk you a little bit about the adults. So in the adults, when we compare black and white individuals, black individuals against black and white individuals, we detect that there are a lot of transcripts that are upregulated in the black and white morph, as you can see that splash on the right of this volcano plot. And we don't see that pattern of the left when we expected to have more probably melanin-associated transcripts. This pattern is clearly seen in this plot. So where at the top you can see a cluster of transcripts that are highly expressed in the black and white individuals, but they are not much expressed in the black individuals. So we decided to make a blast of this cluster of transcripts, and 8 out of 15 are associated with adult-specific cuticle development proteins. And when we plot the counts of these transcripts, we can notice that these 8 transcripts in the black individuals are basically almost absent, and the counts of the black and white individuals are really, really high. So considering all the differential transcripts that we found, we decided to look at the gene ontology of these transcripts. And you can see in the middle there are 4 ontology terms that refer to chitin development, and chitin is a polysaccharide that is highly abundant on the cuticle of arachnids. We also found 1 ontology term that is at the top, specifically linked to cuticle development of spiders, supporting the tendency that the adults' color morph differ in the expression of cuticle-associated transcripts. So to try to understand these differences, the color of the spider can change depending on the chitin or amount of chitin or chitin orientation. So here you can see an example of this spider. So this spider from Madagascar. So this is a microscopic image where the spider is silver. So the silver part has 1 in crystals. And the cuticle is usually transparent, that we observe a similar thing on our dissections. And the layer of cuticle is really smooth. But when you look, here is an image of black part of the abdomen. The cuticle has 2 sublayers. One that is smooth, as is the silver part, and the other one that is where the pigment is deposited. I don't know if this is the same thing that is happening in my spiders, but I plan to do this as soon as I get back to Melbourne. So when we compare the gene expression of the juveniles, we don't find clear difference between the black and white morph, something that I was expecting to find. Maybe a possible explanation for this is that the color expression at this stage may have an hour window of differential expression that was missed in the data that we collected. So I think that I tried to convince you that this is actually a really cool system. Unfortunately, we don't have a genome reference for these spiders, spider genomes, are starting to be sequenced, but we need more. So this system actually offers a great opportunity to test local adaptation, also comparative genomics, and a whole bunch of questions. So I'm at the end of my PhD. If someone wants to talk about this, maybe collaborate, I try to build some projects, I'm down for that. So to conclude, the black and white and the black adults differ in the expression of transcripts associated with cuticle development. As I found with these 8 differentially expressed transcripts that are here. And we did not find differences between the black and white and black juveniles or any difference in melanin-associated transcript as we were expected. I want to thank the Society of the Star Evolution, Ecological Society of Australia, and the University of Melbourne for the support of this research, also my supervisors and the Tiger Lab for letting me use their pipettes. This is it, and I'm happy to take questions.
Very interesting talk. I'm curious if when you were doing the gene expression, you were only taking the carapace off and getting RNA of the carapace, or if it's representing the whole individual. Because it might be that the lack of differentiation that you're seeing is from getting the whole individual rather than just the color section.
Yeah, thank you. That's a great question. So we try to make dissections of the specific part of the coloration, but unfortunately we didn't get a good amount and quality of RNA. So we decided to go with the whole spider. We started them for 5 days to discard any gut content, but yeah, that maybe is a reason why we didn't find the difference. It's all right.
In terms of the ecology of these guys, have you looked at mapping previous fire records with the occurrences that you're seeing? Because there's very few records of animals exhibiting that melanistic phenotype after a fire has come through. And I just wonder if if that's perhaps something relating to that.
You mean like historical records from museums or something like that?
Pardon?
You mean like historical records from museums?
Yeah.
No, unfortunately, I checked all the collections in Australia, and almost all the US are only black and white. There are not many black, so I couldn't do comparative things through time, unfortunately.
So you mentioned the association with precipitation. Can you guess what that is? Why does it make more black spiders?
So yeah, I found the association, but I haven't explored the causality behind it. One hypothesis is that black spider probably camouflage better in humid environments because they are maybe darker, so they get less protected, but eaten. But also it is possible that it has been reported in birds that melanin in humid environments protect against parasites. I don't know. It's something that needs to be explored in depth. Okay, thank you. Thank you.
It's time, so okay. Hi everyone, my name is Tiezheng. I'm a PhD student at the Big Gun Lab from University of California, Davis. So today I'm going to talk about the latitudinal gene expression parallelism between North American and Australian melanogaster and simulants. So first I want to introduce the 2 species that I'm going to focus on today, which are Drosophila melanogaster and Drosophila simulans. So first, they're closely related sister species that only split from their most recent common ancestor around 3 to 4 million years ago, which is a pretty short time period under the evolutionary time scale. And as you can see from the picture over there, they are highly morphological similar. The only difference that you can see is that simulans has larger eyes, and then they have a little hook on their anal pad in the male individuals. And besides that, they also share pretty similar migration histories. So both of them originated in Africa, so melanogaster from sub-Saharan Africa mainland, and then simulans from Madagascar. Later, both species became human commensal and then occupied North America and Australia approximately 200 years ago. However, despite their high level of relatedness, they exhibit strong reproductive isolation that the hybridization seldom happens in the wild. Even if it did, the offsprings are generally infertile. And other differences include that simulants contains more genetic variation, and then melanogaster exhibits clinal differentiation. So in the well-studied North American cline, one of the phenotypic differentiation is wing size, and then genomically, there is the cosmopolitan 3R inversion. So today, my study is going to compare the North American cline and the Australian cline in their adaptation mechanisms in the two species. So we collected wild-type isofemale lines from 4 locations. So in North America, we collect them from Maine and then Panama, which are 2 endpoints of the North American cline. And then in Australia, we collect them from Queensland and Tasmania. And as you can see, the brown line represents the equator, and then the 4 locations are pretty symmetrical across it. And for my study, I'm focusing on the 2 male reproductive tissues, which is accessory gland and testes. The genes that are expressed in these 2 tissues are usually under strong sexual and natural selection, which leads to high sequence evolution rate and the rapid expression divergence, which make them more lineage specific compared to other genes. And the functions of testes is, of course, to produce sperm, and then the accessory gland, which is analogous to the human prostate, produce seminal fluid proteins that facilitate sperm's function. So to capture the expression level of the genes in those locations, we decided to use RNA-seq. So for RNA-seq preparation, we collected 12 lines from each location, and then we dissect the 2 tissues separately from different individuals to avoid contamination. We made 3 biological replicates from per location per tissue. So there are quite a lot of replications made. And yeah, so after we have the result from RNA-seq, we use the lemma, which is a package from R, to calculate the change in expression. So the log-fold changes represents the change in expression from low to high latitude. So a positive log-fold change value represents a higher expression level in the high latitude location. And from the calculated adjusted p-value, we were able to determine the latitudinal DE genes within each continent. So yeah, with the result from lemma, we're able to compare the list of latitudinal DE genes between North America and Australia within each species. But unfortunately, the 2 continents, they don't really share a lot of latitudinal DE genes. Therefore, we decided to focus on the log-fold change values instead, which potentially is able to capture some of the smaller change of expression that might not be able to meet the threshold to be deemed as statistically significant. So here is the result from a comparison between Australia and North American melanogaster in the accessory gland. So as you can see, the x-axis represents the log-fold change values in Australia, and then the y-axis is North America. All the brown genes represents all the brown dots are the genes that are expressed in both Australia and North America. The blue ones are the Australian latitudinal DE genes. And then as you can see, there are 132 of them. We performed a Pearson correlation analysis with them, and then it returns a significant p-values and then a positive correlation coefficient. Now let's go to look at the North American DE genes. So the axes are reversed compared to the previous ones, but again, the backgrounds are the common genes that are expressed in both continents. And then the red ones now are the North American latitudinal DE genes. And then there are more than 300 of them. And then as you can see again, we have a significant p-value and then a positive correlation coefficient. Therefore, we're able to conclude that there is a parallelism in latitudinal DE genes between North America and Australia. And let's look at our testes data. So here we have, again, the axes are the same as the previous ones, and then the blue dots are Australian melanogaster testes latitudinal DE genes, and then again a significant p-value and then a positive correlation coefficient. However, if we look at North American DE genes, we were only able to detect 23 of them, and then only 18 of them were also expressed in Australia. The small number of genes greatly weakened the power of our analysis, which is probably why we're not able to get a positive, significant result. And here is another comparison, but it's in simulants. So this is a comparison between Australian and the North American simulants in the accessory gland. So again, the plot should be pretty easy to read right now. We have almost 300 of them in Australia, and then we have a significant p-value and then a positive correlation coefficient. And then the same with North American simulants in the accessory gland. Yeah, so we're able to conclude that in simulants, there's also a parallelism in adaptation to the geographical variance in North America and Australia in the simulants accessory gland. And after the continental comparisons, we decided to look at interspecific comparisons between melanogaster and simulants within each continent. So similarly, we used the lemma to calculate the log fold change values, and then we used a one-to-one ortholog list between melanogaster and simulants. And again, we decided to focus on the log fold change values. So here is a previous research published in 2015 by Dr. Zhao. The researchers did a pretty similar analysis with North American melanogaster and simulants using whole male body transcriptome. So here are the genes that are latitudinal differential expressed in both melanogaster and simulants. So in the first quadrant, you can see that those are the genes that are upregulated in both species. And then in the third quadrant, those are the genes that are downregulated in both species. And as you can see from the distribution of the number of genes, there's a clear enrichment in the first and third quadrant of them. So we can say that there is a parallelism in the directionality between melanogaster and simulants in North America. And now let's see my result. So this is my accessory gland specific transcriptome, and then those are the genes that are shared in both species in North America, not just the latitudinal DE genes. And as you can see here as well, there's a pretty strong pattern of positive correlation over here. And then if you look more into the distribution of the gene, there are quite a few of them clustered in the third quadrant that is kind of driving the positive correlation here. So I extracted those genes and then performed a tissue specificity analysis using tau values calculated with the data from FlyAtlas2. So FlyAtlas2 is an online resource space that contains whole male each gene's expression in male across 13 different tissues. So it's a pretty good resource for that. So surprisingly, testis biased genes are pretty overrepresented among those genes, which means that they have the highest expression level in testes, which is pretty amazing considering this is a accessory gland comparisons. And next, we decided to look at the physical distribution of the log fold changes along the chromosome arms. So here is a result from here is the X chromosome. So to smoothen the patterns and then to better visualize the trend, we decided to use a sliding window approach. So each dot represents a window containing 51 genes log fold change values, and then each window is one gene with one gene increment. And then the black line represents Drosophila melanogaster, and then the purple one is Drosophila simulans. And then as you can see in the X chromosome, in the midsection of the chromosome, there's a high level of overlap. So not only was the directionality the same, the magnitude also agree with each other pretty well. So to us, this is a pretty surprising and then pretty amazing result, considering there's pretty strong reproductive isolation between the species, like I said, which means that there's little to no gene flow, and then there's short colonization history of North America, which is only 200 years, for about 2,000 generations. So we think that the high level of convergence of changing expression is largely due to natural selection that is acting on them. Yes? So in conclusion, I want to say that like both melanogaster and simulants show parallelism in their mechanism adapting to the geographical variance in the accessory gland. And natural selection has a pretty strong effect in accessory gland transcriptome in melanogaster and simulants in North America. And in the end, I would like to thank my lab for the help and support during my analyses, and especially Dave and Julie for the guidance in analyses, and then Libby, our lab manager, who helped me greatly during the RNA extraction processes. And that will be it. And then if you have any questions, welcome to ask. Yes?
Thank you for the nice talk. It was really nice and interesting. When I listened your talk, I thought about inversions. Do you have information about inversions in your populations, and do they overlap with genes where you can see difference in expression?
So there is a common, like I mentioned, there is a common inversion on the chromosome 3R in melanogaster that is absent in Drosophila simulans. So I use a transcriptome data that is aligned to a pretty standard reference. So my data might not be able to look at inversions. However, we do have a paper that talk about the inversion of it, and then I think I'm actually thinking about aligning the inversion information and then to see if some of the latitudinal DE genes are enriched around the inversion regions. Yes.
Yeah, because it was shown in melanogaster.
Yeah. Yeah. Thank you. Any other questions? Okay. Thank you for listening to the talk.